In this video we will discuss about HIV. So we will what we will discuss about in HIV, how the virus infect the cells and how they are maturated and then get out of the cells and then infect the other cells. We will not go through in details of what is going on within the cells. We will just cover how they are infected and what are the receptors they need in order to uh, enter inside the cells. So let us start the process. This is the HIV virus that enters, considers that it enters from the outside uh, or from another HIV infected patient. And in order to infect the host, it needs to enter into the cell. In order to do so, it needs to the cell needs to have two kind of receptors. One is CD4 and another is either CCR5 or CXC4 CR4. So if these both receptors are present, then the HIV virus can stick to the cell membrane, and then this will further helps the endocytosis of the HIV-1 virus inside the cells. And once it is entered inside the cells, the it has its final location within the nucleus. So it will move towards the nucleus with the help of nuclear pore complex. Nuclear pore complex uh, uh, will help in nuclear transport of this HIV virus within the nucleus. And within the nucleus, as we know that uh, uh, let us consider what are the parts of the HIV so that we can understand this process in a better manner. So this is the envelope part of the HIV, as we know that, and these are the spike protein. The spike protein attaching the helps in making a contact with the cell membrane that which has CD4 and CDC and CCR4 or CXCR4. And after that the uh, envelope interacts with the cell membrane of the host, and that helps in the inner part of the virus, that is nucleocapsid or the core, to come into the cytosol of the cell host cells. And then this capsid, nucleus capsid, then move within the nucleus with the through nuclear pore complex, and this is called nuclear import. And when the HIV virus is within the nucleus, the uncoating of the capsid occurs, and the genetic material that is present within the virus then starts the process of integrating within the host chromosome, and this is called the kind of stable transfection that we have understand in the stable transfection video. And when the host genome integration is complete, uh, then it will use the cell machinery for transcription and translation. The transcription of this modified host genome is when the transcription of this modified host genome occurs, then it will uh, cause the formation of RNA, and this RNA will then export into the cytosol of the cells, where the packaging this genetic material contain packaging portion of the cells and also the uh, genetic material of the virus. So when the RNA get exported from nucleus into the cytosol of the cells, then the packaging and the translation occurs simultaneously. And uh, after that there is assembly process occurs that leads to the formation of new HIV, or the not the formation or the replication of new HIV, or replication of the HIV virus. And when this process is complete, that is packaging, that is assembling of the virus is complete, then the budding process occurs, and this virus then further mature itself, uh, to form, uh, to to get a state that is similar to the virus that when injected, or that when infected the host from any foreign source. And this mature virus then infect the other cells, and this process continue, and the person get infected with the HIV virus as the host, uh, as it is a stable transfection process. So it is difficult, uh, it is difficult for a person, it is difficult to cure a person if the number of cell infected are very large. So so that is the reason most of the peoples are working on preventing this virus to interact or get within the cells. So that is the best approach that people are using. However, for the people that are already get infected with the HIV virus, uh, right now the major approach is to suppress the symptoms of this HIV virus, so that they can live a long and healthy life. So that's all for this video. Thank you for your time.